All right. It is 8, so we'll get started. So as a reminder, next week, Monday, get my pen working here. That is August 3rd. That's 3. The material for test 3 is on the test 3 folder. And I put up that announcement on Wednesday. That just is a reminder that we have some slides that are in the test 2 folder that we didn't get to. That will be on test 3 as well. So the material for test 3 will run through today. So the PowerPoint I have for today's lecture, that will be the last material we have for test 3. Test 3 is going to be same as test 1 and 2, the format, the way it's delivered, all that stuff, where next week, Monday, it will open at 9. Then you'll have until 11 to complete it. So make sure you don't forget that. And as a reminder of what we were talking about last week, Wednesday, we were looking at the membrane of cells, looking at what proteins make up the membrane, looking at lipids, lipid rafts, looking at the types of lipids on each face. And then we ended with this diagram, the secretory pathway diagram, which is a way for the cell to get proteins ready that are supposed to be in the membrane. So remember, these are going to the ER, and they go through this whole pathway to be incorporated into the ER. And then today, we're going to talk about how we can transport these from the ER to the cell membrane. But to begin, we ended on this question, I believe, question 3, where we're talking about mutations. So I have question 4 and question 5 that I think will be good ways to jog your memory, wake you up in the morning. So we're going to start right off with a question here that's a refresher from Wednesday. And I should have a poll for this as well, so I can get those answers. All right. So here's a good example of a test question you might see on Monday. The blank, which is embedded in the endoplasmic reticulum, is a blank protein. All right. So based on what we talked about on Wednesday, see if you can figure out the answer to this. Give you a minute or two to wake up there and see if you can get the correct answer. And we're going to do about 30 more seconds. If you have not answered yet, please do so. Do about 10 more seconds. All right. And let me download those. Okay. All right. So it seems like we're still waking up. Maybe that coffee hasn't hit us yet. The majority of us put B. The ribosome, which is embedded in the endoplasmic reticulum, is a transmembrane pore, which tells me we might not know what a transmembrane pore is. So let's go back to our image. A transmembrane pore is a protein that's in the membrane and basically has a hole through it to allow things to go through it. A ribosome is not a pore, right? A ribosome makes proteins. So it is not a transmembrane pore. The correct answer is C. The translocon is in the ER, embedded in the ER, and it is a transmembrane protein. So if we go back, the translocon in our secretory pathway diagram, that's a protein, and it's embedded in the membrane right here, the membrane of the ER. And you can see it's a pore because it is going through or letting a protein through the membrane. The other popular response was A, that the SRP receptor is a signal protein. SRP receptor isn't doing any signaling. SRP is just binding, or the SRP receptor just binds SRP, and then they hydrolyze GTP to kick the SRP off. And remember, SRP is the protein that stops our newly formed peptide from growing. So step 2 there, polypeptide synthesis is inhibited. So we're not signaling anything. The SRP receptor is kind of just a way to make sure that our ribosome's in place in the correct position relative to the translocon, so that the newly formed protein is going through the translocon properly. And then the GTP is hydrolyzed and the protein is continued to grow. So we're not doing any signaling there. So just as, yeah, so the correct answer there is C. Right, one more question, warm up those brains, get those cobwebs of the weekend out of the way. So here I have the poll again, and I was too lazy to write everything on there because that's actually, it's hard to make a poll like this, but the signal peptide sequence, and you have 4 options. It's often used to direct proteins to the lysosome for incorporation into the lysosome. It's usually not used for small eukaryotic proteins. The signal peptide sequence of some transmembrane proteins embedded into the membrane as an anchor, or is usually cleaved off of the completed protein. So tell me, of those 4 sequences, or those 4 sentences, which ones are right? And A, B, C, D, and E are giving you your options there. And since this is talking about lysosome, I didn't talk about it, but I'll give you a hint. Number 1 is correct. We didn't talk about it in the diagram. We were talking about the ER, but the signal peptide sequence can also direct proteins to the lysosome. So see if we can figure out which ones are here. We'll do about another 30 seconds for this. So please get those answered in. All right, download that poll. There we go. All right, seems like we're up. Our brains the juices are flowing now. So the majority of us said D, that it's often used to direct proteins to the lysosome. Like I said, that's correct. The signal peptide sequence of some proteins is embedded into the membrane as an anchor. Yes, we talked about that, where sometimes that signal is just put right back into the membrane as a way to make sure the protein stays in the membrane. And 4, it is usually cleaved off the cleaved protein. Yes, so both 3 and 4 are true. Sometimes it's embedded into the membrane, but if it's not, it'll be cleaved off. And then we have number 2 is usually not used for small eukaryotic proteins. Well, any protein that is going to the ER, going to the lysosome, it needs a signal. So just because it's small, right, the cell isn't smart enough to know where it should go. It needs that signal. So the correct answer, as the majority of us got there, is D, 1, 3, and 4. Very good. All right. So now what we're going to talk about is how we go from the ER to the plasma membrane. And the way we're going to travel to the plasma membrane is via the Golgi apparatus, right? So after our protein has been transported to the ER and it's finished processing and it's folded, we have to go through the ER to continue our process. And the ER is made out of these membrane sacs called cisternae, anywhere from 3 to 6 different sacs. And we start the cis, ends in the trans. So it's always going from cis to trans. So the cis is right near the ER. The one in the middle is called the medial. And during this movement for the Golgi is when we can do our different types of modifications. So we can start adding sugars, any type of amino acid modifications that need to be done to the protein can also be done in the Golgi. And what's really interesting about the Golgi is that there's different ways to move through the Golgi. If you form a bud, which is being shown here, right? If you just bud off and move from cis to medial and medial to trans, that's called anterograde. So that is anterograde transport, is if you bud off and go from one compartment to the next in like your own little membrane spaceship. However, naturally, the Golgi is like a conveyor belt. So the membrane sac that is in the cis will eventually turn into medial and eventually turns into trans. Excuse me there, I had to sneeze. So the Golgi, like I said, it's kind of like a conveyor belt, kind of like a treadmill, where the different sacs naturally turn into each other. Cis will eventually become medial, medial will eventually become trans. And if you're a protein, you can just hang out in the cis and naturally just be carried all the way to the end. This is called cisternal progression or maturation. So if you are just hanging on and you don't bud off, that's called cisternal progression or maturation. And it is possible to go backwards. That's what this arrow is showing. So if you butt off and you go backwards, that's called retrograde. So backwards is retrograde. So these are just different ways you can move in the Golgi. Antrograde, you butt off. Cisternal progression, you just hang on for the ride until you get off. And then retrograde is if you need to go in reverse. And at the very end, when you come off the Golgi and you travel to the membrane, you're going to travel in a vesicle. All right. So a vesicle is just a membrane sac, a coated membrane sac. And here in this image, it's a little deceiving because we have our vesicle being made out of just lipids. And we're going to see that's actually not the case. Vesicles are made out of proteins. Now, there are lipids here, but vesicles, their shape is dictated by different proteins. All right. And so what we're showing here in the vesicle in this image is we're showing how a protein that is embedded in the plasma membrane actually achieves that. Because in eukaryotes, we don't make our proteins on the plasma membrane and just embed them like that. We make them in the ER, they travel through the Golgi, then they form this vesicle. And so what we're showing here, this purple thing, that's our membrane protein that's going to be in the plasma membrane. These green things, these are carbohydrates. These are sugars that have been added to our membrane protein in the Golgi. Now, the part of the protein that is going to face the outside of the cell, that is in the lumen or the inside of our vesicle. And that is the part that has carbohydrates on it. So right now, before we've even got to the plasma membrane, our proteins in the correct orientation, right? The part that is, well, first off, our protein's already embedded in a membrane, right? It's already in our vesicle. So it's already a transmembrane protein. Remember, a transmembrane protein is a protein that goes through our plasma membrane, and that's what we're showing that this protein is going to become. And so what happens is that this vesicle goes to the plasma membrane and it fuses to the membrane. So we have fusion happening here. And you can see when we fuse, the lumen, the inside of our vesicle, is what is facing the outside of the cell. And this vesicle just becomes part of the membrane. And now we have a new, or two new membrane proteins here. So that is how in eukaryotes, we embed proteins in the membrane. We have them in our vesicles already embedded, then our vesicle fuses with the membrane. Right. So before I continue on, any questions about any of the topics or ideas we have covered so far? All right. Now, like I said on the previous slide, these vesicles are actually coated with proteins. And right now, we know of 3 types of coated vesicles. And the proteins that coat these vesicles are called Clathrin, COP1, in COP2. And the proteins on these vesicles are basically the signal of where they're supposed to go. They're almost like the UPS sticker that says to deliver to this address, right? The protein is saying, okay, if I'm on this vesicle, that means I'm supposed to go to a specific area. So Clathrin vesicles are the vesicles that we just looked at on the previous slide. These are the ones going from the Golgi. So it goes from the trans-Golgi to the plasma membrane. While COP1 and COP2, these are responsible for Golgi to ER, right? So let me just draw a little diagram, as pictures usually help with people. So we have endoplasmic reticulum, Golgi, Clathrin, not Clathrin, plasma membrane. Got ahead of myself. So from Golgi to plasma membrane, that's Clathrin. If we're going from the ER to the Golgi, that is COP2. If we're going from the Golgi to the ER, that's COP1. And a question you might be asking yourself is, why would we want to go from the Golgi back to the ER? Because we've been talking about on Wednesday and today, the process of making these transmembrane proteins is first they go to the ER where they're synthesized. From the ER, they're carried to the Golgi for modifications. Then to the Golgi, they are finally transported to the plasma membrane. However, there are proteins in the endoplasmic reticulum that are supposed to stay in the endoplasmic reticulum. They are not supposed to leave the endoplasmic reticulum. And sometimes, on accident, these ER resident proteins are actually carried to the Golgi. And we don't want them in the Golgi. They're not supposed to be there. So we have a whole system, a whole vesicle protein that their job is to take these proteins that escape the ER and then transform back to the ER. So that's what COP1 vesicles are for. And let's take a look at what these vesicles actually look like. And so on this image on the left here, we're looking at Clathrin, right? And Clathrin is made out of what are called triskelions. It's these like weird looking pre-armed proteins. And here the red would be one copy, the yellow would be one copy, and the green would be another copy. So it's these triskelions that are coming together to form these chains. And they're made out of 3 heavy chains. And now I'm going to jump to the picture on the bottom right here. 3 heavy chains, which are what we're seeing in this microscopic image here. Each of these black outlines is a heavy chain. What we can't see, because they're too small, is that these proteins also come with light chains. So it's not really shown well here, but each heavy chain has 2 light chains. And that's what I've written up at the top. 3 heavy chains, each bound to 2 light chains. And these light chains are a regulatory protein. So when Clathrin has these light chains, it cannot form a cage. It's a way to prevent cage formation. However, when these light chains are removed, then we can form our vesicle chain, cage. And remember, Clathrin are going from the Golgi to the plasma membrane. So these light chains prevent vesicle formation. And then once we remove the light chains, we can form vesicle formation, or we can form our vesicle rather. So that's how Clathrin actually looks like. But let me take a step back here. So we talked about how COP1, and let's just draw our diagram again. So our process that we're talking about is ER, Golgi, plasma membrane. We have COP2, COP1, Clathrin. And the whole reason COP1 exists is to take those ER proteins that escaped and bring them back to the ER. So hopefully the question that you are thinking, or at least the obvious question to me when I hear a phrase like that, is how does the Golgi know which proteins are supposed to be in the ER and which proteins are supposed to be in the Golgi? Or another way to phrase that is, how is the cell smart enough to determine which proteins have to live in the ER? And of course, that answer has to be contained in the peptide sequence, right? Since these are proteins, the only way the protein is going to convey this information is through what amino acids it is made out of. So the primary sequence is how the body can determine, or the cell rather, can determine which proteins belong in the ER and which ones don't. And so in mammals, our ER resident proteins have a specific sequence on the C terminus end, and it's called the K-del sequence. If you're a yeast, it would actually be H-del. But K-del stands for the 4 amino acids: lysine, aspartic acid, glutamic acid, leucine. So these are our 1-letter codes. And any protein that ends in KDEL, or it could be KK anything, or KKK anything, anything, anything. So there's some variation in our sequence, but the sequences that end in this at the C terminus, that's how we determine which proteins are supposed to be in the ER, right? So that's how the cell can differentiate if a protein escaped the ER and is supposed to go back there. So we know that. And then the next question, if you're thinking like a scientist, is, okay, we figured out the sequence that tells us which proteins are supposed to be in the ER. But how do we actually transport these proteins back to the ER? How do we capture these proteins and make sure that the proteins that are supposed to be in the Golgi don't get captured in the process as well? And that's what this slide in the middle is showing. Inside the cis Golgi, we have K-del receptors, and these are proteins. So these are actually proteins themselves that bind to these K-del sequences very strongly. So any protein that is taken from the rough ER and transported to the cis Golgi, if you are an ER resident protein, you'll have this K-del sequence, and then you'll bind this receptor because there's a very strong affinity between the K-del sequence and the receptor. Once you have bound this receptor, you're going to get your COP1 proteins. You're going to form your vesicle, then you're going to be transported back to the ER. And this vesicle is going to form with the ER, and you're going to be delivered back home where you belong. So that's how the cell is actually smart enough to determine which proteins belong in the ER. And this process is how we can deliver them back to the ER. All right, so that was a lot of information on one slide there. So any questions, any clarifications people might have about this process? All right. So let's see if we were paying attention here. This is a multiple answer question, so you can pick more than one. I want you to tell me, these are all C-termini sequences. You tell me which ones are going to be your ER proteins based on what we just talked about. And we'll do about 40 more seconds here. So please put in your answers soon. All right, so let me end that and save it. So these ER resident proteins have the C terminus of K-del or KK, anything, anything, KKK, anything, anything, anything. And if you're a yeast, you also have H-del. So A is not any of those. B is yes. yeast. G-del is not that. K-del is. KCAC is not that. R-del isn't that either. KKEA is. So B, D, and G is what we were looking for there. So any questions about our transport systems here, our ER proteins, or anything up to this point of class? KCAC is wrong because it's not any of these, right? So it's KDEL, KKXX, or KKKXXX. KCAC does not follow that, that system. All right. So we've been talking about how, well, we've been talking about vesicles this last 40 minutes now. And probably in all your biology classes, anytime you talked about a vesicle, you probably just been told, yeah, the vesicle and the membrane just come together and then they fuse. It's actually a lot more complicated than that, because if you remember, the lipid heads are charged. The plasma membrane, those lipid heads are charged. And they're going to be the same charge. They're most likely going to be negatively charged. And negatively charged things don't like to come together. And these things, these lipid membranes, aren't just going to fuse spontaneously. They have no reason to just combine. That's not favorable to do. That's not that spontaneous. It will happen after enough time, but it might take hours or days or weeks or months. And the cell doesn't have that kind of time. So there are various proteins inside the vesicles and on the plasma membrane that help to speed up fusion. And the one we're going to look at are called SNARES. And here we have R SNARES and Q SNARES. The R and Q just stand for the amino acids, arginine and glutamine. There are more SNARES than what we're showing here, but we're just showing a very simplified picture to just get the idea of SNARES down. So what SNARES do is that when we have a vesicle and a membrane coming close together, these SNARES are going to start to zip together. They're going to start winding around each other, becoming tighter and tighter and tighter. And when they start to wind around each other, one, they force the vesicle and the membrane to come together. And two, you're also increasing the curvature of the membrane. And by doing this, you're doing a couple things. One, you're helping to overcome the charge repulsion from the vesicle and the membrane. You're forcing these to come together. The second thing that these SNARES also help with is to remove water. So keep in mind, we're in the cell, right? Between this vesicle and this membrane are tons of water molecules. And if we want this fusion to happen, these water molecules have to get out of the way. And this snare, when we bring the vesicle and the membrane together, we are pushing the water out of the way because we're forcing these things to come together. And so the water has nowhere to be. And so it'll be moved to the side. So we're getting rid of the water between the vesicles. And as these things zip up, zip up, zip up, you're going to start to see some fusion happen now with the membrane. So this empty square here, that is actually showing that these lipids are being removed. So we have to remove like the contact point lipids if we want to fuse these vesicles together. So those are removed. And then we can have our fusion happen, right? And our snares are going to continue to twist and twist and twist until, as shown in this image right here, in the middle bottom, our vesicle is now part of the membrane. They are one continuous membrane. And so here, this is delivering proteins that are supposed to, or molecules that are supposed to go to the outside of the cell. And so that is what we have done. We have now delivered our molecules to the outside of the cell, and we just have one continuous membrane. So that's what the snares are really doing here. They are making sure our vesicle and our membrane come together and that they fuse. Now, inside the cell, this whole process takes about 0.3 milliseconds, incredibly fast. If you try this process in a test tube with just snares, you're looking at 30 to 40 minutes. So there are a lot of other proteins that have to be involved with this process. So it's just not snares. There's going to be a lot of other helper proteins, but we are just looking at snares here of how to combine the membrane and pores, or vesicles rather. So yeah, any questions about this diagram people might have? All right. Well, of course, I have a question to make sure we understand this then. So let me get this pull up. So which one of these sentences best describes snares? So is it 1, 2, 3, or 4? Read those over and put in your answer, and then I'll be back in a few minutes to discuss. we'll do about 20 seconds for this. So please get those answers in. All right. And the correct answer, as the majority of us got it, since we were just talking about it. So thank you for paying attention there. It is 3. They overcome the electrostatic repulsion between, wait, they do not do that. They do 3. The wording is a little funky there, between vesicle molecules. It should be between vesicle and the plasma membrane. Just realize that now, but yeah, the best answer there is 3. They are not. Let me just go over why the other ones are not wrong or are wrong. Essential for proper post-translational processing. That's happening in the Golgi. SNARES aren't doing that. Critical element in the function of the secretory pathway. The secretory pathway is happening in the ER, and so the SNARES aren't doing that. Facilitate anchoring between vesicle membranes. Not really. They are there to really help to overcome that repulsion we talked about between the different lipid membranes. So 3 is your best answer. And just a couple more slides here. So we're talking about the plasma membrane, talking about SNARES, and the idea of SNARES to overcome that electrostatic repulsion. Viruses have something similar. So here we're looking at flu, the flu virus, how it attacks our cell. COVID uses like the same mechanism. And in fact, the vaccine for COVID, what it's attacking is basically this system. It attacks the spike protein, which is the protein that more or less latches on to your membrane and like punctures a hole into the membrane. And so that's what the vaccines are targeting. And that's why so far the vaccines are good against all these variants that we're seeing, because the spike protein is the same in these variants. And as long as that spike protein doesn't wildly mutate, the vaccine should still be good against all these other variants. And just as a friendly reminder, if you have not gotten vaccinated yet, go and get vaccinated. Unless you are like immunocompromised, you should definitely get vaccinated. But yeah, let's look at flu hemagglutinin here. And what this is a protein that is going to help the virus attack your plasma membrane and pull the virus in to overcome the electrostatic repulsion. And it's going to work like a jackknife. And in case you don't know what a jackknife is, it's kind of like this knife that I'm showing down here, where it will be folded. The blade will be inside the handle. And then when our reaction happens, this knife will spring out. And we have a similar thing happening here, where we have our protein and it's looking for salicylic acid. And we talked about how on our plasma membrane, we have certain lipids that have salicylic acid. And so flu is looking for these lipids. And when we find this lipid, the hemagglutinin is going to bind to it, and then it's going to jackknife, which in essence pulls the flu towards your plasma membrane. And then since the virus is being pulled towards the membrane, we are going to overcome the electrostatic repulsion. We're going to fuse our viral membrane with the plasma membrane. Then we can dump our RNA into the cell, and we can make trillions of copies of ourselves using using the cell's ribosome. But yeah, that is how the flu, that is how COVID can infect cells. They are looking for a specific marker on the outside of the cell. Once they bind to that marker, the protein that is doing that binding is having a conformational change. That conformational change brings the virus in the plasma membrane closer in contact, which is overcoming that repulsion of electrostatics, allowing the virus in the plasma membrane to fuse, and now you have an infected cell. So the idea of SNAREs, we also see that in viruses as well. That's the main take home here. Now, the last thing we're going to talk about for this PowerPoint before we get on to the new one is math, everyone's favorite subject. We're going to look at the thermodynamics of diffusion through the plasma membrane. And if you go to this section in your book does an awesome job of walking through all the terms and why these terms were chosen. But if you don't learn well through looking at equations, I know some people do. The only way they can learn math is seeing how it's worked out step by step, like how do you get this equation? If you don't, if you can just look at an equation and you learn better by just using it, that's fine. And the equation we are going to be using to determine the energy of movement is this equation, where delta G equals RT, so R is the gas constant, T is temperature, natural log of the concentration of molecule A in its final location divided by the concentration at its initial location, plus, and this is supposed to be a lowercase, Z of A, the charge of A, and Z of A is something that students seem to get confused on a lot. So let's imagine that we are moving chlorine. Chlorine has a charge of -1. So Z of A for chlorine is -1. Sodium has a charge of +1. So Z of A for sodium would be +1. Magnesium is +2. So Z of A for magnesium is +2. So it's just saying, what is the charge of your molecule? That's all Z of A is. So plus the charge of your molecule multiplied by Faraday's constant, so just a constant, 96,485, multiplied by the electrical potential of your membrane. So the final charge minus the initial charge. So inside the cell and outside the cell, these are not neutral environments. They have charges. And so if you're moving ions or a molecule that is charged, you have to consider, am I moving to a region of positive charge or am I moving to a region of negative charge? Because that affects the thermodynamics. That affects the energy of movement. So we're going to use this equation to determine is the movement of certain ions spontaneous or not? What is the total energy of movement for ions here? So let's take a look at this equation in action. So sodium potassium pump, and I'm a big fan of drawing pictures when it comes to word problems. So let's draw out a picture here. So the sodium potassium pump transports 2 potassiums into the cell and 3 out of the cell. All right, so we have a cell here and we have a pump. 2 potassiums are going into the cell. 3 sodiums are going out of the cell. And inside the cell, the membrane potential is a negative 100 millivolts. Now, membrane potential. What that is saying is it's saying, what is the difference between the charge of the inside the cell and the outside the cell? That's membrane potential. What's the charge difference? Here, we're saying the inside or the membrane potential is minus 100 millivolts. So the membrane potential is saying, what's the difference between the outside and the inside? If the inside is negative 100, we're just going to say the outside is 0. All right, so I know historically, again, that assumption confuses people. So I'm going to say it one more time. The membrane potential, that is just saying the charge difference between the inside and the outside of the cell. In total, the membrane potential is minus 100. And we're saying, well, the inside is the one that's negative. So to make the math easy on myself, I'm going to say the inside has a charge of minus 100 and the outside has a charge of 0. These numbers could be anything, as long as the difference between the two are negative 100. Like I could say the outside is 1,000 millivolts. And I could say the inside is negative 1,100 millivolts. It doesn't matter, as long as the difference between the two is minus 100. So to make it easy on myself, I just do 0 and negative 100. So that's membrane potential. Now let's continue. The extracellular fluid, so the outside of the cell, has 140 millimolar sodium, has 5 millimolar potassium. The cytosol has 10 millimolar sodium and 100 millimolar potassium. What I want to know is, what is the delta G for the movement of 2 moles of potassium and 3 moles of sodium? This is spontaneous. Do this at 37 degrees C. Here's your gas constant you're going to use. Here's your equation, and there's Faraday. So I'm going to give people like 5 minutes to struggle through this. And I do want you to try and figure this out on your own. Please don't sit here for like 5 minutes and not do anything and just watch me do it at the end and just think to yourself, okay, I saw it done once. I'm going to know how to do it from there. Really, the best way to learn is by doing. So try your best to do this. See how far you can get and try to come up with the answer. And then once you see my answer, see what I did versus what you did and see if you can figure out, okay, where did I go wrong? What didn't I see there? But yeah, I'm going to give people like 5 minutes to try and do this problem. If you have questions during this, please send them to me in chat. Otherwise, let's see if we can do some math. All right, so hopefully you had some time to struggle with that. Hopefully some hands-on experience to figure out what this equation is saying, put in some variables. Let's take a look at how to do this. So we have two different ions we need to consider. We need to consider sodium and we need to consider potassium. So let's do sodium first. So let's look at this equation. So the delta G of movement for A, well, in this case, it's Na because it's sodium, is R, gas constant, 8.314, times T. Here, our temperature needs to be in Kelvin. Because if we look at the gas constant, our gas constant has a unit of Kelvin, and I gave you a T in Celsius, so those don't match, so we need to convert. So 37 plus 273, which is 310. So R times T times the natural log of A final. That means the concentration where you're going. So for sodium, we are being pumped outside the cell. So our final concentration is 140. Our initial concentration, we were being pumped initially from inside the cell. So our initial concentration is 10. So this is the natural log of 140 divided by 10 plus the charge of sodium, Za. So that would be the charge of sodium is 1 multiplied by Faraday's constant, 96,485, multiplied by the membrane potential. So this is final minus initial. So our charge in our final location is 0. Outside the cell, our charge is 0. Minus the charge of our initial concentrate, our initial area, which is negative 100. So this is 0 minus a negative 100 millivolts, which is positive 100 millivolts. So this is saying that the sodium is experiencing a charge difference of 100 millivolts from being moved from inside the cell to outside the cell. Now, one more complication to throw on there. Faraday's constant is in volts. So we can't be in millivolts, we need to be in volts. So here, our change in membrane potential is 0.1 volts. We do that math. And that should be 16,450. $16,450 joules per mole. So that's the energy for moving 1 mole of sodium. However, I'm asking for 3 moles of sodium. So you just take this number, multiply it by 3, right? Because it's 3 moles, not 1 mole. And the energy, whoops, that's not what I want. The energy of moving 3 moles of sodium outside the cell under these conditions is 49,350 joules. All right, before I move on to potassium, what part of that did people get confused on? What part of that equation right there, for the numbers I put in, did I lose people? Is there anything you want me to repeat or re-go over for those sodium numbers? right, since I'm not seeing anything pop up, let's do potassium then. So potassium. Same idea, R 8.314 multiplied by T, same temperature, so 310, multiplied by the natural log. So for potassium, it's going to an area, the final concentration is 100. And initially it was coming from an area that was 5. So that's natural log of 100 times 5. Plus the charge of potassium is 1. So 1 times Faraday's constant multiplied by the charge difference. Here, our final destination is negative 100 millivolts minus our initial location outside the cell is 0. So here, it's actually the opposite sign of sodium because we're going from outside the cell to inside. So it makes sense, it would be the same number, but opposite sign. So this is multiplied by negative 0.1 volts. Needs to be in volts because Faraday is in volts. So we do that calculation and we get negative 1,927 joules per mole. So the movement of one potassium is negative 1,927. However, we have 2 moles of potassium. So I multiply that number by 2. And that's negative, that should be a 2, not a 3. That is negative 3,855. I just forgot that number I just said. Negative 3,855. So that is the movement of 2 moles of potassium. And since this is a process that happens together, you just combine those numbers. So the total delta G for this process, for 49,350 joules from sodium minus 3,855 joules from potassium. And the total energy of this movement is 45,495 joules. Not spontaneous. This is a non-spontaneous process. All right, now that you saw potassium worked out as well, what questions do people have? What clarifications do people want? Where did I lose you? Anything at all? Or did everybody just get 45,495? Easy peasy. Will you only be working with sodium and potassium when you use this equation? No, you can use any molecule in the universe for this equation. And the only thing that changes is Z. What is the charge of the molecule? No, this can be used for glucose. This can be used for bananas. It's not sodium and potassium specific. Do we need to know their direction of movement? Yes, if you do not know which way they're moving, there's no way you can figure out the membrane difference in membrane potential. There's no way you can figure out the concentration at the final and initial locations because you don't know the final and initial locations. So yes, you would have to know like which way we're moving to be able to do this calculation. Is it non-spontaneous because this is secondary active transport? It's not secondary active transport, but it is active transport, which is actually the next question I always ask after this. If this is not spontaneous, yet it happens in your cells all the time, how does this happen? And it's not secondary, it's primary. So what does primary active transport mean? How do we make this spontaneous in the cell? How does this actually happen inside of all your neuron cells, inside all your cells every single day? Will we be given a question where we are asked to solve for a missing variable for this equation? For the most part, for this equation, you're going to get a problem that's very similar to this one. So no, I won't ask you like, what is the final concentration if delta G is this? I could, but I won't. I would expect people to be able to do that, but I'm not going to ask you to do that. But yeah, let's get back to the question I just asked there. How does the cell make this non-spontaneous process actually happen? Because those concentrations I listed, that charge differential I listed for the membrane potential, those are real conditions inside your body. And that's the direction the sodium potassium pump works. 2 potassiums in, 3 sodiums out. So how does this actually occur if I just told you it's not spontaneous? Nope, osmosis is only water movement. We input energy with ATP. Yes, exactly that. When we break phosphates off ATP, we release energy, and we can use that energy to power a non-spontaneous process. So yes, you would have to use ATP to make this pump work, which is what happens. Right. So with that, before we end this PowerPoint, any other questions about anything we have covered today? All right, so let me get up the PowerPoint for today. Share screen on 7/26. And we're going to continue talking about movement of ions, which was the equation we just did. And there are different ways that you can move ions through the membrane. One's called an ion 4. Well, they're both called ion 4, but the difference is between a carrier and a channel forming ion 4. And the question here is, why do we need to have like proteins to move ions across the membrane? So let me ask that to begin with. Before I talk about ion 4s, why is it necessary to have a protein to transport ions across the membrane, right? Ions are small, right? Why can't ions just go through the membrane themselves? Why do we need proteins to help move ions across the membrane? Because the membrane does not like the charge. Yeah, basically, right? Remember that the inside of the membrane It's, well, yes, let me rephrase that. The ions don't like the membrane. The membrane doesn't care, right? So remember, the membrane here is nonpolar. There's no water in there. Ions are surrounded by water. And it's very, very energetically unfavorable to strip all that water away and not surrounded by anything but nonpolar residues. So unfavorable that it will like never happen. So it's the ions that don't like the membranes. The membranes, they're fine. Remember, nonpolar things are fine interacting with polar things. Oil has no problem interacting with water. It's the water that doesn't like to interact with oil. So here, it's the ions that don't like interacting with the membrane. So two different types of ways to bring ions across. A carrier will bind the ion and then travel through the membrane and then deposit the ion on the other side of the membrane. That's called a carrier. While a channel is a transmembrane protein that just has an open pore that allows ions to go through. Both these methods speed up ion diffusion a lot. And these carriers and channels, they're usually only specific for one type of ion. They only cater to one type of ion. Like valine-myosin here, valine-myosin transfers potassium and only potassium. It doesn't do magnesium. It doesn't do sodium. It only does potassium. So what we're going to look at is we're going to look at some of these channels in the time remaining today. So one of the channels we're going to look at is for maltoporin. And maltoporin is a part of these class of molecules called porins. And a porin is a beta barrel with an aqueous channel in the middle. So a beta barrel is shown here. Whoops, you cannot see that color. All right. So it's beta sheets that form a giant barrel. And in the middle is our aqueous chamber. So the outside, so these white lines I'm basically drawing here. So this would be the membrane. So this porin is embedded right in the membrane. And in the middle, we have a pore that's allowing our sugar to go through. And when we talk about all these different pores, the same theme is going to come up again and again and again. These pores are specifically designed to allow only one type of molecule through. So let's look at our maltoporin here. So our maltoporin has all of these yellow residues. And these yellow residues are aromatic residues, right? These are our big rings, right? Our tyrosines and our tryptophans and our phenylalanines. These rings interact with the ring structures of our sugar. Because remember, our sugars that we talked about in the sugar chapter are also made out of rings. So we are interacting through our pi stacking there. Our rings are interacting, which allows our sugar to move through the channel. We also have a bunch of amino acids that will hydrogen bond with all of the hydroxyls of our sugar as well. So what happens is that our maltoporin comes in and it travels like a screw through our pore, right? These aromatic and hydrogen bonding residues direct it like a screw through our pore. And this type of setup only allows maltoporins through. Other sugars cannot use these specific transporters. they're specific for one type of sugar only. So that's a porin. Porin are beta-barrel structures, and our multiporins only allow these maltodextrins. In case you've never heard that word before, dextrin is just a fancy way to say sugar. So it's allowing these malto sugars to go through our channel. It does it through these aromatics and hydrogen bonds. And just FYI, let me undo that. The questions I like to ask about this section is, the question I usually ask is, relate the structure and function of, for example, multiporin. So don't be shocked if you see on the test, like, explain how the structure of multiporins help it with its function. And the way to answer that is, well, the function of multiporins is to allow multidextrins to go through the membrane. And so the structure that helped with that is that they have a bunch of aromatic residues to interact with the ring structures of the sugar. They also have a bunch of residues that will hydrogen bond with the sugar that will allow it to move through the aqueous pore in the maltodextrin. So that's the kind of understanding I'm looking for. Can you relate structure and function of these channels? So any questions about that? What I'm looking for, or anything about the multiporin specifically? All right. And the next type of pore we're going to look at is the potassium channel, KCSA. And the potassium channel, of course, transports potassium. And there are a lot of different potassium channels, but they all have similar sequences. And basically, they are 4 subunits that form a pore. And that's what's being shown in this middle picture, where the pore is right here in the middle, and these 4 different proteins, well, 4 copies of the same proteins are coming together to form this pore. And the way that we make sure potassium, only potassium, is transported through this pore is that the pore itself changes diameter, right? It goes from 10 angstroms all the way to 3 angstroms. And the idea with that is at the top, we have the 10 angstroms. That's because our potassium is going to have a bunch of water molecules around it. So it has a bunch of water molecules around our potassium. And so the top of the pore can accommodate that. It accommodates potassium with the water molecules. However, as we shrink, then our pore, our potassium rather, sheds those water molecules, because we don't want water to travel with the potassium. We only want potassium. And so as the pore shrinks, the water is removed. So water goes bye-bye. And now our pore is shaped so that only potassium can fit. So let's imagine that like a sodium tried to get in here. First off, at the top, sodium and all those water molecules don't have the right dimension, so it probably won't bind at the top. But even if it did, the sodium is not the right size to go through the channel down here. So sodium does not get transported. But the pore is perfectly sized to allow our potassiums to go through here. And as you can see on this image, these red things, they are oxygen molecules, right? And so this pore is just covered in negative charge, which is shown right here as well. So we have negative charge at the top and at the bottom of our pore. This will attract the potassium to bind. In the middle of our pore, though, let me get a different color for that. In the middle of our pore, we have yellow. So that is showing we don't have negative charge in the middle of our pore. Again, that's actually a very clever idea. At the top, we have negative charge to bind. However, this non-negative charge in the middle forces our potassium to go through the pore fast because it does not want to interact with things that aren't negative. So when it's in the middle of the pore, it wants to get out. And so this will propel it to move through our channel faster to this negative charge at the bottom. And once it's down here at the bottom, water can reassociate with it again, and then it'll be transported inside the cells. But that is how potassium channels work, right? We have variable diameter pore, bunch of oxygens in there to allow only potassium to go through. So any questions about that explanation of the potassium channel? All right. So here's a question. One of the diameters of the potassium channel is roughly 6 angstroms. I want to know why a single water molecule has a diameter of 2.75 angstroms. Why can't that go through the potassium channel? If the potassium channel is 6, wouldn't it make sense for something smaller to go through? But that doesn't happen. Does anyone have can anyone verbalize, make sense of why that could not happen? In the videos we watch, you talk about proton hopping. Is that why? That has to do with aquaporins. So not here. We'll talk about proton hopping when we get to aquaporins, but that's not the reason why here. Does that have to do with polarity? Kind of. So let me draw a picture that might help. So remember, I talked about how the potassium channel has like a bunch of oxygens at the beginning of the channel. And if we just have one water molecule, it is too small to really interact with this channel well. And since it's not interacting with this channel, it has no reason to be here. Energetically speaking, it is unfavorable for water to just hang around with nothing surrounding it. Because water is a polar molecule, so it wants to hang out with other polar molecules. So if a water molecule finds its way into this channel, well, it can't interact with the channel, and so it's going to pop right back out because it's not getting what it needs energetically. But if we're talking about potassium, which is something like that, and it would be surrounded by water molecules at this point, but potassium being solvated would be the perfect size to interact with all these oxygens. So really, these channels are more or less a story of Goldilocks, right? A water molecule would go in there and say, this channel's too big. I can't go through it, so I'm not going to. While a potassium would go in there and say, oh, this channel's just right. It fits perfectly for size and charge. I'm happy, so I'm gonna go and interact with this channel. And so that's actually the idea of all these channels we're going to talk about. It's basically Goldilocks. They have been created to only incorporate one molecule. So that's why something smaller can't pass through it. It doesn't interact with the channel. And so it's not going to pass through the channel if it can't interact with it. So questions about that idea? Okay. So let's see here. Just seeing where we're at. All right, let's get through the nervous system then. So your nerve cells, the way they work is through using these ion pumps. And they actually use two different ion pumps. They use a sodium and potassium channel to propagate a signal. And the way that they open and close their channels is through a change in voltage. So these are called voltage gated channels. So what happens is that your neuron receives a signal from your neighboring neuron. And when that happens, a sodium channel opens. So a sodium channel opens down here. When a sodium channel opens on your neuron, that allows sodium that was on the outside of the cell, that just rushes into your neuron. And what happens then is that the charge of your neuron changes. So let me refresh this slide because there we go. And you can see it, that's kind of what we're showing here. So I'm gonna wait till it gets back to the start. Okay, at the start, signal, sodium flows in. When sodium flows in, the inside of your neuron becomes positive. So inside becomes positive. Outside is now negative when the sodium flows in. And this change in charge allows a second voltage gated channel to open, and that's the potassium channel. So sodium flows in, the inside of your neuron becomes positive, and potassium opens. And when this potassium channel opens, potassium flows out. And this is called hyperpolarization. So much potassium flows out that this becomes way more positive. And this causes another sodium channel to open up in right next to your potassium channel. So sodium flows in. This causes your potassium channel to open up. So potassium flows out. This causes another sodium channel to open up. And it just repeats over and over and over again in your neuron. And this is how we propagate a signal. So let me again restart that image to show. So signal, and you can see how the charge, the positive charge, is just moving across the neuron. That's these sodium and potassium channels opening and closing. So the way a neuron is able to transmit a signal all the way through it is through charges and through opening and closing these potassium, sodium charges. And the whole process that we're looking here is called an action potential. We can visualize this action potential on a couple different graphs. So we're going to actually look at the B graph here to begin with. So after about 0.8 milliseconds after receiving your signal here, your sodium permeability skyrockets, which means sodium channel open. And it closes very fast. You can see this sodium channel is not open for even a full millisecond. So the sodium channel opens, sodium flows in, and then it closes. And the sodium flowing in makes your potassium open in response. You can see how there's an overlap where first the sodium is at the peak, and after that, your potassium starts to open as well. This will cause your potassium to flow out, and then the potassium channel closes. And we can look at that through action potential graph. So these two graphs, the y-axis is the same. So our action potential happens. We get that signal, opens up the sodium. The sodium opens up the potassium. When the potassium flows out, our membrane potential goes below where it was. So at time 0, this is the charge inside the membrane. It's at -60. After the potassium leaves, it goes to -70. So it actually takes some time before your neuron goes back to its -60. It takes about, looks like it's taking about 3 to 4 milliseconds before your neuron is reset. And when the sodium is open, your cell goes to like +30. So it goes from -60 to +30. That's huge change. And that is how your neuron works. It's just a sodium and potassium channel that are opening, closing throughout your whole membrane. And then when we get to the very end, we can send our signal to the next neuron to continue our message. So questions about this sodium potassium channel system here? All right, so it's 9:48. So I'm going to say this is the last slide for test 3. Anything past this, you're going to see it on the final as new information. But since this is where we got to on Monday and your test is a week away, this is where we're going to call it. So just make a note on your notes about how anything past this will be on the final. Yeah. Other than that, we'll call it a day. I'll put homework up as always. Your tests next week. Don't forget that. Study hard. If you have any questions for this week, feel free to email me. Feel free to stop by. Other than that, hope you have a good rest of your Monday. And I will see people later. Any last minute questions people might have before I sign off? If not, since I'm not seeing anything pop up and I'm seeing people leave, have a good Monday.